Thank you.
Thanks so much. It's great to be here. Thank you for inviting me. Thanks to the organizing committee, and thanks especially to Linda for putting this in perspective and getting us all together. So what I'm going to do today is talk to you about the interrelationship between fat, muscle, and bone. And I'm going to focus on brown fat, particularly because there's been a lot of recent studies that support this interaction as being quite critical both to the skeleton and to other organs, rather than talking about fat per se, which you've already heard some discussion about in the other two talks. So I'm going to amplify a little bit about what Stephanie mentioned about the nervous system, and as you can see from this diagram, the sympathetic nervous system is a major player in regulating skeletal function, as I'll show you, but also in terms of browning of white adipose tissue, and certainly in terms of muscle. And I also will amplify a little bit on what Tammy said in the last talk about sedentary lifestyle and the role of neurotrophic factors in enriched environments, including brain-derived neurotrophic factors. So this is a physiology talk, basically, and I'm going to try to emphasize for you how important it is not to think about a single tissue, such as muscle, or we do in terms of bone, but in terms of perturbations that occur in one system are going to have major effects on other systems as well. And this sort of outline just briefly describes this potential interaction between brown fat and bone, which I'll allude to. Gerard has done a lot of work on showing that the skeleton has a major effect on white adipose tissue, insulin sensitivity, as well as insulin production. And of course, white adipose tissue's interaction with the hypothalamus and central nervous system via leptin and other mediators. And I want to emphasize in this talk, and one of the central tenets of this talk, is the function of PGC1 alpha, PPAR gamma, coactivator 1 alpha and beta, which turns out to be one of the central components of the integration of muscle, bone, and fat. So I think everybody in this room is familiar with the concept that in the bone marrow, and certainly in the stromal vascular fraction and elsewhere, and probably in satellite cells, that bone, muscle, and fat all arise from the same stem cell, this multipotent stem cell, which is a progenitor which can differentiate into the osteoblast or adipocyte lineage, or certainly into a muscle cell. And we all have done experiments showing we can do that in vitro. It's a little less clear what happens to these brown-like adipocytes, particularly in the bone marrow, and I'll come back to that shortly in my talk. I do want to make one point clear, and I think you'll see this when we talk about the browning of white adipocytes, is that we're still not clear whether there is transdifferentiation that occurs between a more mature progenitor going from fat to bone, and certainly for the white adipocyte in becoming a brown-like adipocyte, as you'll see on the next slide. So I think we focused a lot on understanding that there is a progenitor cell that can give rise to these three mature cell types. I think it's less clear, and it's one of the questions that I raised at the end, about the metabolic hierarchy that also occurs. And this is where it's really remarkable how far we've come in the past 5 years, particularly in the adipocyte field. And that is, of course, we've known that white fat is a storage depot for fatty acids and triglycerides, and myocytes and muscle tissue obviously use energy, substrates, glucose, and fatty acids to generate ATP, as well as for motor function. But this intermediate, the brown fat, or the preformed brown adipose tissue, and a brown adipocyte, which we've known existed in the interscapular region for some time, is a strong intermediary. In fact, it comes from a MIF5 progenitor, which also can differentiate into muscle cells. So this is sort of a hybrid between a muscle and a fat cell, and I'll explain that in a second. Less clear, much less clear, and a question that we really have to sort of tackle, but it's not easy, is what is the bioenergetics of the osteoblast, and what substrates do they utilize, how do they utilize it, and is that machinery critical in terms of differentiation into a bone cell rather than a fat cell? So we've known for a long time that brown adipose tissue exists in mammals, particularly hibernating animals, and its major function, probably its only function, is thermogenesis, and that is the uncoupling in the electron transport system via the mechanism of uncoupling protein 1, which allows for the generation of heat rather than ATP generation. And the hallmark of the brown adipocyte, which we see in the interscapular regions, are lots of mitochondria, small fat cells. They look brown. If you open up a mouse and examine the interscapular tissue, it is brown. And if you look at some mouse models like we have in our lab, where inducible brown adipose tissue is occurring due to a high sympathetic tone, the color of the tissue is brown rather than white. And that's more mitochondria and the expression of PGC1 alpha. Whereas, as you're familiar with, the white adipocyte is a storage depot. It does not express UCP1. Fatty acid oxidation is much less. And indeed, as I mentioned to you, the key hallmark for brown adipose tissue is uncoupling protein 1, which uncouples protons away from ATP generation and transport into the inner mitochondrial matrix with the generation of heat. And the question that our lab has been focused on now for the past couple of years is understanding the relationship between brown adipose tissue and the skeleton, and of course the other converse association, the association of brown adipose tissue, which has a mitochondrial phenotype that is reminiscent of muscle, and its relationship to adult muscle. So just to remind you that there's now been fairly good data from the group at Quebec, as well as data in mice, that the principal source of heat thermogenesis in brown adipose tissue occurs with fatty acids, both in humans and in mice, as I mentioned, and again, this beta oxidation, which leads to heat generation. Now, it's only been in the past couple of years that we've grown to appreciate that besides preformed interscapular brown adipose tissue, whose hallmark is the MIF5 progenitor, there is a brown-like adipocyte which exists in subcutaneous tissue. It exists in the retroperitoneal tissue. It's more prevalent in certain fat depots than others, but under the influence of sympathetic tone, these cells, if they originate from stem cells or if they originate from white adipocytes and transdifferentiate, can become brown-like adipocytes and can generate heat. And this browning of white adipocytes, you can imagine the potential therapeutic implications of converting white adipocytes to brown adipocytes. And this has exploded the obesity field. And I'm going to focus on what those implications are for the skeleton as well as for muscle. This is just some work from our collaborator, Les Kozak, showing back in 1999 that preformed brown adipose tissue differs by strain in mice, not a surprise, but more importantly, there are differences, and we're certain that this happens in humans as well, in the amount of inducible brown-like adipocytes that are present in other fat depots. And in mice, the inguinal fat depot is particularly susceptible to induction of brown-like adipocytes. So one of the questions we asked early on is, how does brown fat or bright fat relate to bone and muscle? So when I talk about bright fat, that's brown-like fat. They call it beige. Bruce Spiegelman now calls it beach. I mean, there's a number of different terms, but it's somewhere between white and brown. So if you can think about that as the inducible brown adipocytes versus the preformed brown adipocytes, which we have in our interscapular region, that'll be helpful. It gets to be confusing. So there are some clinical data. This is work from our laboratory with Miriam Bradella at the Mass General that we just recently reported, showing that the volume of brown fat as measured by glucose uptake using PET-CT is directly related to bone density. And then, you haven't seen this yet, but I just accepted this for publication, obviously a bias of the editor, but this is from Vicente Gilsans, also showing that the volume of preformed brown adipose tissue as measured by PET-CT is directly related to bone mass, and this is cortical mass. Now, the nice thing about the Gilsans study is that when you correct for muscle mass, you lose the correlation with bone density. So, not surprising if they're originating from similar progenitors, that the brown adipocyte, the preformed brown adipocyte, may be related to muscle. And then there's a paper that's just finishing edits from the group at the NIH, also showing a strong positive relationship between bone density, areal bone density, and the volume of preformed adipose tissue, but only in women, not in men. So what other examples do we have? Well, this is work from the Davis group at Baylor, published several years ago, but it's quite remarkable. They take BMP2 overexpressing cells, they put it on muscle tissue in mice, and then they watch for heterotopic ossification, and you actually see the full range of endochondral ossification. So you get vascular invasion, you get cartilage development, and you get skeletal development with marrow in these sites that are in the muscle. Now, the interesting thing about this is that at day 1 of the BMP2 overexpressing cells, you see these fat cells which disappear. And indeed, whoops, so that's not right, but we can get out of that. Indeed, what you see is UCP positive cells that exist the first 24 to 48 hours of this process of heterotopic ossification, suggesting that the presence of fat cells, some fat cells, cells that express UCP1, may actually be important for osteogenesis, and we'll come back to that a little later on. Finally, this is work from our colleague Beata Lekis-Zernick. She took the FOXY-C2 transgenic mice. So the group in Sweden overexpressed FOXY-C2, which is a transcription factor for brown fat. They overexpressed it in fat cells using the AP2 promoter, only in fat cells. And what they found was tremendous increases in insulin sensitivity and increases in lean mass. And Beata's going to show at ASBMR that there's also significant increases in bone density, as well as increases in marrow fat, which are expressing UCP1 positivity, suggesting that there is this direct relationship between brown fat and bone. And she will describe some of the mediators that are released from brown adipose tissue that may be important in enhancing osteoblast differentiation. Indeed, as also noted, the individuals, mice that have the FOXY-C2 have increased insulin sensitivity and increased lean mass. So one of the things that's happening in the obesity field are challenges to change white fat into brown fat. And this has been something of a novel series of experiments that have been going on in research laboratories, but also in pharmaceutical companies, because obviously if you take a pill or a drug and you can get brown adipose tissue, you can lose weight, burn off energy, this would be spectacular for the obesity epidemic. So we already know some of these. Actually, the TZDs, if you look at gene expression, the TZDs, Rosen and PO, actually do increase both PGC1 alpha and PPAR gamma activity and have brown-like adipogenesis in peripheral depots. More recently, FGF21 has been considered a major browner of white adipose tissue. Unfortunately, as Dr. Wan showed at UT Southwestern recently in PNAS, FGF1 overexpression actually causes bone loss. And this is probably through the activation of PPAR gamma. So you're beginning to see the story that PPAR activation, which may be good for browning of white adipocytes, may actually not be good for the skeleton. Beta agonists such as isoprel have long been shown to have a positive effect on adipose tissue, but a negative effect, as Gerard has shown, on the skeleton. I'm going to mention ricin, and I'm going to talk about two other components, the enriched environment syndrome and cold exposure. So just to remind you, PPAR gamma is at the center of this whole story. And initially, PGC1 alpha was actually described as a coactivator of PPAR gamma in brown adipose tissue. But as we know, PPAR is trifunctional. It is important for adipocyte differentiation. It enhances insulin sensitivity. And it is an important stem cell fate determinant. In addition, it is also important, obviously, for the browning of white adipocytes. So PPAR gamma is necessary for adipocyte differentiation. It's the recruitment of the co-activators and the co-repressors that help determine what type of adipocyte is present. And it's PGC1 alpha that, when it's recruited to the receptor complex, actually will enhance the generation of UCP1, whereas in white adipocytes, PGC1 alpha is not active and co-repressors are present. So what's going on with PGC1 alpha and beta? It's a multi-purpose coactivator for fatty acid oxidation, angiogenesis, electron transport assembly, and mitochondrial DNA function. So this is one of the central features of mitochondrial bioenergetics. And it can bind to a number of nuclear receptor complexes. I just mentioned the PPAR-RXR heterodimeric complex, and we know that that's important in fatty acid oxidation. More recently, PGC1 alpha has been shown to bind to estrogen-related receptors, including ERR1, which has been shown by Dr. Wan's group at UT Southwestern to be important in osteoclastogenesis. And as I mentioned, PGC1 alpha is a critical factor in electron transport chain and in the generation of mitochondrial DNA. So several years ago, about 3 years ago, there were a couple reports using the muscle overexpressing transgenic, the MCK-TG, for PGC1 alpha. And in that report, they found increased muscle mass, not surprising, but they also found high areal bone mineral density, and it was not described in any greater detail. But in addition to that, what they also saw was this browning of white adipocytes. So these animals, not expressed in muscle, still had this fat phenotype, which showed tremendous increase in mitochondrial content and a browning associated with UCP increases, as well as SIDEA and a number of other factors, suggesting that PGC1 alpha had a significant role at 3 levels, tissue levels, that is certainly muscle, bone, and in this case fat as well. Now, nobody really understood how PGC1 was active. We know that it's upregulated during exercise in muscle, and the question was, what was it doing and what downstream were the activating compounds or molecules? And it was left to Bruce Spiegelman and colleagues in a very important Nature Medicine paper just recently, who identified a secretory peptide that was induced by PGC1. So the first thing they did is they overexpressed PGC1 alpha, again in muscle tissue, and looked downstream at what factors were upregulated. And certainly one of the big ones was FNDC5, which is it's a membrane protein which hadn't really been described in great detail, but was present in muscle tissue primarily. And it turned out when you overexpress PGC1 alpha, you see all the changes in browning that you would expect. But as Spiegelman showed, you could recapitulate the changes with FNDC5 when you looked in the circulation and identified this cleavage peptide of FNDC5, which is irisin. They called it irisin for a Greek goddess. This is a peptide fragment of the parent compound on the membrane, which is cleaved during exercise and induces this browning, which is identical to the overexpression of PGC1 alpha. And several studies have now shown from the Spiegelman lab that this is associated with increased insulin sensitivity, upregulation of the brown-like markers, UCP1 and SIDEA, as shown. And suggesting that this is a major factor in exercise-induced muscle changes leading to fat changes. What we don't know is what irisin does to bone. So remember, I set this up by saying some of the agents that we think about, the brown-white adipocytes, have a negative effect on the skeleton. We don't know what the bones from those animals look like. So exercise induces PGC1 alpha, which stimulates this membrane peptide FNDC5, which is cleaved, although we don't really understand what the enzyme involved in that process is. Irisin circulates. It converts white adipocytes to brown adipocytes. So this has huge potential from a therapeutic point of view, if indeed this is validated in other studies. Now, I want to focus on the central nervous system because Stephanie mentioned that we need to think about it, and Gerard has been a pioneer in understanding how the sympathetic nervous system regulates the skeleton. And we do have a lot of data, mostly from his group, but from others as well, showing that the sympathetic nervous system has a negative effect on the skeleton. But this is work looking at another neuromodulator, and this is the group, the cow group from Ohio State. And what they did is they noted that when they put mice in an enriched environment, so remember what Tammy said. Tammy said that we're all sedentary now and we don't do anything. Now, could that have a negative effect independent of everything else that's going on? So they asked this question, and they put mice in a playroom, a little play cage, and they looked at the effects of the enriched environment on bone, and this on muscle and on fat, not on bone. And it's a great paper. Everybody should look at it sometime because it's really remarkable. So they knew that exercise induced brown-like adipogenesis and insulin sensitivity. But the question was, did this enriched environment with play tools in the mouse's cage have an additional effect? And what they found was, indeed they did. This is retroperitoneal adipose depot, and you can see when the mice were subject to a nice environment with slides and tools and everything else, and they played a little better, they actually had browning of white adipocytes. It was actually quite a remarkable story. And the mechanism is actually quite interesting, and it's mediated through PRDM16, which is a cofactor that is important to suppress the differentiation of white adipocytes and to enhance the generation of brown-like adipocytes. So this is an interesting story, and we should be thinking about sedentary behaviors as having a central effect. And they were able, using a AAV vector, they were able to actually introduce BDNF, brain-derived neurotrophic factor, into the hypothalamus, and that does upregulate sympathetic tone. So sympathetic tone is a major mediator of this effect. So we asked, what happens to sympathetic tone in terms of the skeleton? And Gerard had already shown that it has a very negative effect on bone turnover. RUNX2 is suppressed, and rankle is increased. So we have been studying two inbred strains of mice. This is the C3H mouse and the black 6 mouse, and we've mapped genetic determinants for these very high bone mass versus a much lower bone mass. But what we failed to see until recently is that their thigh muscle area was also significantly greater in C3 than it is in B6. So they have high bone mass and greater muscle mass, and more importantly, their grip strength is significantly greater in C3 than B6. But the other remarkable thing about C3H mice is they have tons of marrow fat consistently throughout their lifespan, always more than B6. And in the marrow, they have a gain of function, which we previously reported, but we didn't understand the implications in JVMR, a gain of function in PPAR gamma. So they actually have increased expression of PPAR gamma in the bone marrow compared to C3H, about 5-fold increase. When we expose these animals to cold temperature to induce acute sympathetic tone, what we found was B6 mice don't do anything, but C3 with a gain of function in PPAR actually have this remarkable increase in both UCP1 and PGC1 alpha, suggesting that at least we think in the bone marrow of C3 that they're expressing a brown-like adipogenic program, which is thermogenic and may actually be enhancing the effects of the skeletal increases that we see. Okay, I'm going to finish my story with just a little bit on the absence of preformed brown adipose tissue. So one of the things about aging that nobody talks about is that we lose brown adipose tissue, our preformed brown adipose tissue. We used to think we lose it at 2 years of age and after infancy, but we now know we can see it on PET scan. But it definitely goes away with age, and we can show in the mouse by 38 weeks of age that brown-like fat cells have now become white-like. So they're no longer performing their thermogenic program. So we wanted to know if that had any effect on muscle and bone. So we looked around for models. We have the UCP1 null mice, and they have terrible bones, but they're also a difficult model to work with. So we've latched onto the Misty mouse. Misty has a loss of function mutation in a gene called DOX7, and it was described back in the late 90s to have absent brown adipose tissue. So we have been studying that, and indeed, what DOX7 is, loss of function mutation, this is a spontaneous mutation, is DOX7 is very important for axonal direction. So it helps in terms of generation of the axon and its direction. But what we didn't know until recently is that DOX7 is highly expressed in both muscle and bone cells. And DOX7 is phosphorylated by ERB2, which is also upregulated by a number of factors that people have used in skeletal studies. And downstream, it's a Rho GTPase, so it activates RAP1 and CDC42 and ultimately junk. So this is a very interesting mutation, and we've identified one of the biggest problems with the brown adipose tissue is the lack of neural sympathetic innervation of the brown adipose tissue. So this is tyrosine hydroxylase staining, and you can see that there's virtually no staining for sympathetic nerve fibers in the brown adipose tissue. So how did these animals survive? They've been around since 1942 at the Jackson lab. If they're not functioning with brown adipose tissue, how do they survive? And they survive because they got tons of bright adipose cells. So if we look in the inguinal tissues, we see a tremendous increase in UCP1, PGC1 alpha, and if we expose them to cold temperatures, so we put them at 4 degrees for 6 hours, they are able to control their body temperature by the induction of these brown-like adipocytes. More importantly and interesting, when we feed them a high-fat diet, these animals remain quite lean. And again, this is an increase in sympathetic tone. Unfortunately, when we expose them to cold, they also drop their RUNX2 and importantly have a marked increase in rankle expression. So again, this sympathetic tone has a very important role in regulating skeletal mass. And you can see the changes by micro-CT, which are more progressive with age, and we suspect that's increased sympathetic tone. And this is data using propranolol to try to rescue the skeletal phenotype, and we're able to show that at least we get partial rescue. We certainly get rescue with trabecular thickness and total body BMC, and partial rescue of trabecular number just by giving them 4 weeks of propranolol in their water. However, as is always the case in bone biology, nothing is as simple as that. And certainly, we now have identified a cell autonomous defect in osteogenesis. These cells don't attach well, and we think they're misdirected. Their cytoskeleton is misdirected by the DOX7. I want to finish by just mentioning that we've done energy expenditure studies, and we initially got a little confused because we expected to see higher energy expenditure. These are metabolic cage studies with the Misty mouse. And actually what we found was they had lower energy expenditure at 8 and 16 weeks of age. And this led us to ask a very important question. This is lean mass versus total energy expenditure. It said, what else is going on in this animal? And what we found was that their abdominal muscle mass is very much less than in their litter mates, and indeed other muscle function studies suggest that they not only have low bone mass, but they have low muscle mass, which may be a cell autonomous defect. So I'll finish by just reminding you about, I think, some of the key principles that I've mentioned. First, PGC1 is a critical cofactor linking, we think, linking bone, muscle, and fat through mitochondrial function. The central nervous system, and particularly the sympathetic nervous system, plays a key physiologic role in bone, fat, and we believe in muscle interactions. And factors for muscle, such as irisin, may enhance thermogenesis centrally or peripherally, but we don't know what it does to the skeleton. And brown-like adipocytes may have a positive effect on bone through secretory factors that Beato will show at the ASBMR meeting. that actually may not only stimulate muscle function, but may also stimulate osteogenesis. And we think MISTI is a useful model. So the questions that I wanted addressed included 3. Is age infiltration of muscle with fat a similar process to age-related marrow adiposity, which is what Tammy asked? And if so, what's its origin and function? And we think marrow adiposity is very context-specific, and what we see in C3H may be very different from age-related marrow adiposity. One of the big holes in our field are the bioenergetics of bone cells, and how do these relate to mitochondrial function in muscle cells, and particularly in bright cells? And finally, and this has been very difficult, and we've been trying to do these studies, what is the role of the sympathetic nervous system in age-related muscle atrophy and in bone loss and in marrow adiposity? So this work's been kindly supported by 3 institutes, and this is our lab group in Maine. So thank you very much for your time.
There's time for 1 or 2 questions for Cliff, and then we'll move on to the discussion.
Okay.
Particularly San Francisco. Cliff, that was great.
Thanks.
But you've confused me.
Yeah.
So we have a sympathetic nervous system, which, as you point out, Gerard has pointed out, is not so good for bone, and yet it seems to be good for brown fat. You showed us data that I think you're concluding the brown fat is good for bone, but we've always thought at least the white fat was not good for bone.
Right.
So where are we here?
Well, I think that's the problem. We don't know. I mean, I think the preformed brown adipose, our hypothesis is the preformed brown adipose tissue prevents the induction of inducible brown adipose tissue in other sites. In other words, if you have thermogenesis, then you don't need to activate the sympathetic nervous system peripherally. And we think that activation peripherally has a deleterious effect. So there's a proposal in Maine to actually lower the temperature in classrooms by 2 degrees centigrade in kids to see if we can reduce the rates of obesity. And our collaborator, Les Kozak, has a thermosensor, infrared sensor, so we can actually measure brown adipose tissue heat generation. So that's our hypothesis, that as long as you have preformed brown adipose tissue, you're not activating that compensatory mechanism.
But the sympathetic nervous system is going to activate that brown,
right, that's right, that's right.
Fat.
But what's really remarkable is Dan, just let me just finish that, is that we get inducible brown adipose tissue in the MISTI mice, even though the function of the preformed tissue is not appropriate, suggesting that there are differential neural regulation, and that's what we're trying to get at.
I guess the thing is a little bit different than, because you have brown fat, but what the sympathetic nervous system does is to brown the white fat. That is good for the white fat, but is also bad for bone. And this is a conundrum that everybody in the leptin field, for instance, has worked upon, is that yes, they can lose weight, but because you give leptin, you also increase the sympathetic tone, you lose bone. So the idea that the sympathetic tone is good for the brown fat is not accurate. It is good for the white fat because it browns it, but that is bad for bone.
Right.